Go ahead. All right. Do you see the presenter view or the regular?
Yes.
You see presenter view? No, you're
Yes.
good. Oh, we're good. Okay, great. Yep. All right.
Oh,
Well, perfect. Thank you so much for that introduction. Great. So I'm Holly Stessman. I'm in Department of Pharmacology and Neuroscience here on the Omaha campus. And so today I'm excited to talk about genetics. I say that genetics is kind of my love, my medical genetics. And the topic that my lab works on is autism spectrum disorder, and that's really derived from the work that I did as a postdoc, which I'll kind of touch on really briefly because it kind of leads us to where we are now. So just a little outline. I'm going to talk very briefly about autism spectrum disorder and how kind of the genetic and clinical evidence that we have for a particular gene we're interested in the lab called KMT5B. And then we'll talk a little bit about how we're using a mouse model to try to understand that gene a little bit better. And that's going to include some kind of ongoing data collection that we have and maybe get a little feedback about the direction we're going. So broadly speaking, a lot of my work has worked in just neurodevelopmental disorders across the board. And so that includes things like autism, but many other things as well that share some kind of common core clinical domains. So things that affect things like executive functioning, language development, social skills, and also learning and memory. And so while a lot of what I'll talk about today is focused on autism, which we lovingly call ASD, we also work a bit in so these other comorbidities that often track with autism. So things like intellectual disability, motor delays, things like that. But one thing that really drew me to this field is this idea that, particularly in autism, we understand there to be a really strong genetic component, which is kind of what drew me to this field from working in cancer previously. So we've come a really long way when it comes to understanding genetics and autism. So when Leo Kanner first described this disorder in the early 1940s, it was thought at that time actually that parents showing a lack of warmth for their children is what was causing autism. So they kind of referred to this as refrigerator mothers. So we've come a really, really long way from this. And that's driven primarily by twin studies that started in the 1970s that really started to show that there was a higher frequency of concordance in twins of autism diagnoses than you'd expect by chance. And so the current studies, what we currently understand in autism is that probably somewhere between 62 and 75% is genetic. And that the rest is environmental and other kinds of contribution. And that's really been driven by these large twin studies that have been performed in the last 50 years. So with that being said, a lot of the work that I did as a postdoc was really trying to identify those genes that contribute to that genetic side of the house. And, it would have been great if there were one or two variants that caused autism. That would have been easy, a home run, low hanging fruit for us. But unfortunately, or maybe fortunately for the field, as far as having stuff to study for the next, 100 years of lifetimes, really we found that probably somewhere in the order of 500 to 700 genes are involved in autism and just variably kind of contribute to this really complex genotype phenotype relationship. So our work that started, kind of around, 2012 or so when next gen sequencing really came on board has worked to kind of contribute to the amount of genetic data we have. And so this is just to show a recent study, which incorporated some of the data I worked on as a postdoc, but really is the most comprehensive, I would say, meta-analysis of whole exome data that we have currently in autism. And so because we know autism is genetic, one of the models we've decided to utilize in studying autism is what we call a sporadic autism model. And so on the left-hand side, just to kind of show it as a pictorial representation, we look for families where you have two unaffected parents that have an affected child. Or multiple affected children. And this idea here is that, there's something present in the children that's not present in the parents that's causing that autism phenotype. And so if we sequence all three of the individuals in this kind of trio structure, we can try to identify genetic variants that might contribute to autism. And so that's what kind of this big study is showing is multiple kind of foundation and NIH funded efforts to do exome sequencing on these trios. So these data represent about 12,000 trios or so. of individuals with autism. And you can see that, there's a number of genes that are rising above the white noise. And so specifically, our current count is that we believe we have a really good handle on about 100 genes that are involved in autism, but this is still ongoing. We still need more samples, with a magnitude of number of genes of the size that we're talking about, it's going to take a lot more sequencing. And so,
Holly,
yeah.
sorry, I hate to stop you so soon, but so this is really curious, of the like 25% or whatever that aren't thought to be genetic, I assume that's like what the sporadic sample set is, but they're still genetic. It's not some environmental influence during development or something. It's a genetic mutation or alteration that leads to basically all autism. Is that what the
So
thought is?
So actually the genetic contribution, so like the twins, for example, that would include sporadic cases. So you're just
Okay.
talking about, we certainly see cases of familial, right? So things like kind of run in families. So really kind of out of the genetic component, which would include inherited genetic variation and sporadic, we're really only looking on the sporadic side because we don't understand the inherited forms very well and especially how they're different between males and females and how that presentation works and how that inheritance works. So
Wow.
this is out of that kind of genetic side of the house, really not taking into account the environmental potential impacts. It's just the sporadic side of the genetic side.
So most of the genetically linked ones are sporadic.
Yeah, they are. We
Wow.
think they are.
That's so interesting. Thank you.
So I didn't include it here. I actually took it out at the last minute, but one of the things we're finding that's really interesting is that, in the past, in other types of genetic disorders, like things like Down syndrome, aneuploidies, it's assumed or has been shown that the age of the mother really affects that outcome. So
Mhm.
interestingly, in autism, and they're seeing it more and more in others as well, we're finding that the age of the father actually really matters a lot. And probably the sporadic variants that are SNPs or single nucleotide variants are more likely to come in on the male germline, and copy number variants and aneuploidies are more likely to come on the mother's lineage. So it's really interesting because fathers kind of were without fault for a long time, and we definitely see this signal that's highly repeatable that the age of the father really contributes to SNP acquisition, basically, in offspring. Yeah, really interesting. So yeah, when I was kind of finishing my postdoc, and still this is ongoing, right? There's big, computer science and mathematics groups that are working on this, obviously. And as a function person coming from the cancer world, I was always the one in lab meeting, raising my hand saying, yeah, but what do these genes do, right? And I think that's kind of where we're at in the field, is that computation only takes us so far. So we can use things like, trying to group people by genes and phenotypes that they share to try to understand if there are genes that are in the same pathways, or, looking at where we think these genes might be expressed in the brain using publicly available data sets like the Allen Brainspan Atlas to try to understand, are there genes expressed in certain tissues in the brain over time? And even trying to think about cell type enrichment now that we have single cell data sets, thinking about, are these genes represented in single cell populations that might explain the biology? But I would argue that the computation in mathematics is kind of reaching this limit of what we can understand about what these genes actually do and how they contribute to biology. And so that's where I decided I wanted to kind of work on making my mark, was try to, take individual genes and really hone down in what these things are actually doing. So my lab really has focused on a gene called KMT5B. It was previously called SUV420H1, which is a mouthful. And that's because it was identified actually originally in the suppressor of variegation screens in flies, where they were looking for epigenetic modifiers that could affect variegation patterns in the eye. Obviously, this is going to be an epigenetic modifier that I'll talk about a little bit more in a minute. But we identified this gene first when I was a postdoc, identified several patients, saw this gene starting to come above that level of significance, and started to think about what it might be doing. And I will point out that this larger study that just came out a couple of years ago, they are repeatedly finding this signal coming out in the big data sets too. So this gene seems to be withstanding the test of time, and we've become more and more interested in it. So I'll tell you some about our stories. So our really big kind of overarching question in the lab is, how does KMT5B specifically contribute to autism diagnosis? And so a little bit about what this gene does. So it's an epigenetic modifier. And so specifically what we mean here is this gene and the methylation mark that it lays down is thought to regulate chromatin compaction. So actually what we find is that this enzyme lays down methylation marks at the lysine 420. So it's on histone 4 lysine 20 position. And that increasing methylation at this particular residue leads to increasing chromatin compaction. So in this family that methylate this residue, there's KMT5A, B, and C. So we're talking about B, the one in the middle, which is thought to lay down a dimethylation mark using a monomethylated substrate. So KMT5A is thought to lay down the monomethylation, then B does dimethylation, then C does trimethylation. And we think this happens sequentially. And this uses a SAM methyl donor group in this reaction. So what we know about kind of association with compaction is that the more methylation you have, so increasing methylation is associated with chromatin compaction and therefore gene silencing. So that's the main function of this enzyme that was kind of known before we started all of this. And a lot of the work was done in yeast and in Drosophila a little bit as well. But so we're kind of focused on this dimethylation kind of step, but we're actually coming to understand now that there might be some other side functions of KMT5B specifically involved with the dimethylation mark. And one of those, which has obviously piqued my interest because I am interested in cancer as well, is this idea that dimethylation at this residue is involved in the process of recruiting a repair factor called 53BP1 that helps to repair DNA double-stranded breaks through homologous recombination. So this 53BP1 actually directly competes with BRCA1 to repair these. So obviously there is, some potential for implication in cancer as well, which KMT5B has a little bit of literature associated with cancer biology as well. But that's not the primary function I'm going to talk about today. It's more the epigenetic kind of side of the house. So one of the first things we kind of did on this project was to ask, some of the broad questions, right? Is this gene expressed in the right place at the right time to, potentially be involved in brain development? So we use the Allen Brainspan Atlas, which is a collection of, wild type brains. So people who died of natural causes that span all the way from 8 weeks post-conception all the way through several decades of life. And so they've actually microdissected all the regions of those brains and then done RNA-seq on each one of those regions. So we looked just at kind of the average of that signal for all the genes in this family. So specifically KMT5B here is going to be in orange. And we see actually that the most expression, the highest levels of expression actually occur during in utero development. So embryonic development in humans. In the brain, right? And so this would be overlapping processes like neurogenesis, some gliogenesis, a little bit of early synaptogenesis, but kind of leaving out some of these other processes, suggesting that there might be some obvious places or processes to look at when we're thinking about what the function of this gene might be. And then when we look across actually all those regions, since I said we have that kind of regional specificity based on this data set, maybe fortunately and unfortunately, we find that our gene is very broadly expressed across almost all regions of the brain during in utero development. So I say this is good because it means probably our gene's doing something important if it's constitutively expressed across lots of regions. But unfortunately, it doesn't really give us any key regions to think about or look at specifically of where this might be really key or essential. And also, as you might not be surprised to hear, I'm going to talk to you about our mouse model a bit today. So just to show you kind of in the wild type developing mouse. So this is embryonic day 14.5. This is during kind of early peak neurogenesis time. So it would be kind of equivalent to this window I'm showing you here in human. We do see that KMT5B, which is shown in brown here through DAB staining, is really it's very broadly expressed across a lot of the body with really high expression in the brain at this time as well. So we're lucky that KMT5B has a homolog in the mouse and that it does seem to have an expression pattern that's relatively similar to what we can glean from human literature. So we had a recent study, thinking about if we really wanted to robustly go after specific phenotypes associated with KMT5B, that we needed to better understand the patient population. And so this was a collaboration that just was published a few months ago with my collaborators at CHOP, so the Children's Hospital of Philadelphia, where kind of our two groups decided to put our heads together and recruit as many patients as we could that had known KMT5B clinical or research mutations. and look at them, look at their phenotypes, right? See if there's anything in common that we could test in our mouse. So this just is a picture of some of our cute kiddos, and then showing you where those mutations fall on the protein structure. So you can see here, maybe not surprisingly, a lot of the mutations that are expected to affect protein function. So this would be putative loss of function mutations and missense mutations that affect protein and are predicted to affect structure. Most of them do fall really close to the known protein domain, the set domain, which is the domain known to actually perform the methylation kind of enzymatic activity for KMT5B. They do seem to pile up in this area of individuals who are affected. So we took kind of a broad sweep of phenotypes of these kiddos. So did some deep clinical phenotyping, reaching back out. And I will say almost all of these data are retrospective. So the physicians went back and dug into those records and, said, can we see any evidence of things like, hypotonia, like low muscle tone, things like that. So they really kind of dug for very specific traits for us to see if there was any evidence in the patients. So in black is a yes indicator for these patients, white is no, and gray is they were unknown. So they couldn't find any information related to that domain. So you can see here, we've kind of divided our patients into different groups. So we have males versus females, and then putative loss of function mutations versus missense mutations. And really we divided based on sex because we know in autism, there's a significant sex bias, about 4-to-1 males to females. So we wanted to look at those populations also separately to see if there were differences in severity. So some things I kind of want to point out. So here's our 43 patients shown by patient number on the left-hand side. Some things I kind of want to point out. So one thing that we definitely saw was pretty much everybody in our study had a diagnosed intellectual disability or developmental delay, something significant enough to make the diagnostic criteria. And that was kind of interesting to us because most of the patients that had come in before actually came in on autism studies. And actually autism prevalence was pretty low across our population, lower than we would have expected. I think it was about 56%. So whereas intellectual disability and developmental delay was in everyone. So that was kind of interesting to us because we expected it to be a more prevalent autism gene than an intellectual disability gene. So that again, maybe bears some reassessment in using our animal models. The other thing that was interesting was macrocephaly was a really strong finding in our cohort as well. So macrocephaly is basically a measurement of the circumference of the head. And it's something that's done in well child visits pretty much across the board. So it's a really easy measure to go back and look for. And the reason that they use it a lot in the autism literature is there has been some imaging studies that have shown that early brain overgrowth might actually be involved in autism diagnosis. But the interesting thing about this is that, and I'll get on my soapbox for a second, is that macrocephaly in the research community for autism is often assumed to be indicative of a brain overgrowth phenotype. However, we know from imaging studies that head circumference and brain volume are only correlated between the ages of like 2 and 8. And after that, they're no longer correlated. So this is potentially problematic because it means that we don't understand whether this has to do with the brain itself or whether it has to do with just overgrowth of the head. And so this is something that we are particularly interested in. We also saw in this cohort, there was a statistically significant sex bias for macrocephaly toward males over females. And so that is also kind of interesting because macrocephaly in males has been shown to be repeatedly a neurophenotype associated with autism in males. So that is kind of in agreement with those data. The other thing that was interesting that we looked at was whether this was associated with tall stature. So all the reports that came before this one have had maybe like 1 to 3 patients. And so they've been very small kind of collections of patients. And several of them reported that their patients that had macrocephaly also had tall stature. So we wanted to know whether this was actually a broad overgrowth phenotype because interestingly, for those who are on the call who work in the cancer world, mutations in PTEN actually are found in the autism population, and they are associated with a significant overgrowth body phenotype and also increased cancer prevalence. So because it is obviously a cancer gene. So there is some evidence that those two things track together. But in our population, we actually found that that wasn't the case. So we were able to kind of debunk some of those early studies. So these patients have macrocephaly, but it is not associated with tall stature, suggesting that it is head specific in some way. The other thing we found that was really interesting that'll come up again is significant hypotonia phenotype. So this is a low muscle tone. Sometimes they call it floppy baby syndrome. The babies have trouble kind of holding their heads up and getting coordinated movement. This was pretty significant in our population. It was not what we expected, but was really exciting based on some of the skeletal muscle data I'm going to show you in a little bit here. And then finally, we saw pretty significant facial dysmorphology in our patient population, but there was no consistent face shape associated with this gene like you would expect with things like Down syndrome, where you expect a very specific facial dysmorphology. So with that said, we decided, kind of in tandem to use our mouse models to see if we could recapitulate any of these patient phenotypes with this idea that if we knock down expression in the mouse, that they may show features that are similar to the patient population that we could then interrogate further. And so we use a gene trap mouse model. This is from the Mouse Knockout Project. For those that are not mouse savvy, you can essentially think of this as knocking out the copy of the gene, either on one locust or two, depending on how you do your breeding strategy. For those who are mouse savvy, this is a gene trap allele, which essentially inserts an early stop polyadenylation site upstream of the functional domain, which causes nonsense mediated decay and loss of that transcript on that allele. So essentially, however you decide to understand it, it results in a knockout of the allele. So the first bit of...
You do have a question in the chat, Holly.
Yeah, oops.
What were the ages of the kids at the time of collection or of the study? Were there fewer kids diagnosed with autism compared to developmental delay because they were too young to diagnose?
That's a really great question. Yeah. So the ages were very broad because they came kind of from all over the world. So a lot of the data that we had was age at last contact of visit. From what I understand, I think from what I remember, there was maybe only one person under the age of 2, but their current diagnostics are pretty confident in diagnosing individuals over the age of 2. But I have to say, diagnostics have come a long way in better identifying autism in girls, which is a little bit more challenging than in boys. So it's hard to say about whether age played a factor in that. I'm sure sex played a factor in that. And then also, the ascertainment piece, we do always worry about, whether we get more or less autism than expected. Generally happens based on how the people present and which clinic they come to first. So if they're coming because they are severely intellectually disabled, they're going to be assessed mostly for intellectual disability genes, versus if they're coming in for autism, they might be assessed for that because whole genome sequencing isn't CLIA-CAP approved, but panels are. And so lots of times the ascertainment on what type of clinic they came into and what kind of provider they were seeing could guide what type of patients we get. And that's certainly, the ascertainment bias is something we can never discount. Great question. Thanks, Joss. All right. So the first bit of work that I'm going to show you is kind of the early phenotyping we did in this model. And this was by 2 really talented individuals that were have since left my lab and moved on to other positions. But the first was a really great postdoc, Rochelle Wick-Brahmasekara. She actually was a PhD student in BMS here at Creighton and then moved on to do a postdoc with me. And then Bryn Robertson was actually a really great postbac who worked for me for 2 years before she joined the PA program. And she's going to graduate from here in December with her PA degree. So both of them were really fantastic and worked to collect the data in these kind of papers I'm going to show. So the first little bit of kind of testing that we wanted to do in the model was to think about, okay, well, what's happening in the brain of these animals, right? We do think that KMT5B is being expressed in the brain. What genes is it affecting? So we collected RNA from the brains of embryonic mice at that kind of time point where neurogenesis is happening. And we, in fact, could validate in our model that we can knock down the expression of this gene. Great, that's a positive control. The other major kind of broad finding that we found was that there was an inverse correlation between KMT5B expression and the expression of the monomethylation state. And so that's kind of interesting. We expected to see more effect on the dimethylation state since that's the direct target of KMT5B. So that suggests to us then that based on this model, right, that if you knock out KMT5B, that the dimethylation, something else is rescuing that, either KMT5B or something else, but it does cause a reversion or at least a pileup of that monomethylated substrate. So that's interesting to us, particularly from the chromatin compaction and gene expression side of the house, because it suggests if this state is favored, then you would expect there to be lower chromatin compaction and more gene expression. And in fact, that's what we see. So if we do RNA-seq on those same brains, we do see that there seems to be a global upregulation of gene expression, even in the heterozygous state, that we also see in the knockout as well. So that's kind of interesting to us. And so obviously we are interested in what genes live in this camp, right? What genes are being upregulated and significantly downregulated, and does that give us any more information about gene types that might be specifically affected or pathways? The other bit of data we kind of overlaid with this, we were lucky enough from our patient population to have fibroblasts from a few of those patients that carried KMT5B mutations. I think we had maybe 6 or 7 lines that we could get to grow robustly. And so we actually did gene expression on those as well. And compared them to our mouse data set. So some caveats, right? The amount of overlap in these is pretty small, but we're talking about fibroblasts versus brains and all kinds of different, genetic background in the humans versus our mouse, which is genetically controlled, right? But we do see some overlaps, which is really exciting. So we do see overall that genes that are upregulated in human samples, there's a more global gene upregulation. It's a little bit more muted than what we see in our mice, but it is there. And there are some overlapping genes that have given us some kind of food for thought for moving this project forward. But we used our mouse data set to further actually think about what kind of pathways might be implicated based on our gene expression data. So this is an analysis from Ingenuity pathway analysis, and it's showing from our mouse up and down regulated gene sets taken together, pathways that may be up or down regulated. And so some things we got particularly excited about were that there were a lot of brain pathways and mechanisms on the inhibited side of the house. So in blue here, we see a lot of potential, prediction of changes in neuridogenesis, axonogenesis, development of neurons. So for us, this seems like, there might be a morphology and or growth issue going on with some of those populations that we've been thinking about more. Some other things that are interesting based on like the hypotonia phenotype that we were seeing, we see some predicted activation of genes that are involved in movement disorders. So thinking about whether, that actually might be contributing to the hypotonia phenotype in our patients. The other kind of bit of phenotyping that we decided to start with our mice was actually to do a behavioral battery. And so the most common question I get from people in research who have worked with mice, but not in the autism side of the house, are how do you know if a mouse is autistic? Can a mouse be autistic, right? So the short answer is no, we've never seen evidence of natural autism in a mouse. However, we're really lucky that mice are actually social creatures. They like to live in groups. They don't like to be isolated, and they also crave novelty. So they have some similar behaviors, I guess, that are things that we could develop tests against that might be relevant to autism social behaviors. So this is our mouse battery that we started with. We looked at a number of autism-related tests that have been kind of discussed in the field and developed in the field. We also looked at some learning and memory tests to kind of look at that comorbidity, and then a number of other comorbid phenotypes that are related to like anxiety and things like that in our mouse model. And importantly, we tested male and female mice. We had cohorts of both because of that sex bias that we potentially see in the patients. So this is to show you an example of one of our autism-like tests. This is a social preference test. So basically, it's a little 3-chamber test. The test mouse is placed in the middle, and on both sides are placed mice under these cups here. And the mouse is allowed to basically interact for a fixed amount of time with whichever mouse they prefer, for however much time they prefer. So generally what we do for this test is that we place a cage mate on one side, so a familiar animal, and we place a novel animal on the other side. And you would expect in a wild type mouse, so a typical mouse, that they would want to spend more time with the novel mouse than they do with the cage mate because they prefer novelty. And so what we see in this test is specifically in females that the wild type mice are behaving the way that we expect. So they want to spend more time with the novel mouse, but this novelty seeking behavior is lost in our heterozygous animals. So I will point out on all the tests I'm going to show you today, you're only going to see wild type and heterozygous animals. And that is because the knockout is perinatally lethal. So shortly after birth, the knockout animals succumb we don't know why, actually. We haven't really been able to figure out why they die. But again, kind of, contributes to this idea that this is probably an important gene. So overall, kind of from our social studies, we found that females more than males seem to have these social changes. And also, I didn't show here, but they do have repetitive grooming behavior. So they do the same sequence of kind of repetitive over grooming, which also is kind of an indicator of autism-like repetitive behavior phenotype. In addition to the behavior tests, we also looked at a number of kind of reflex development things. So just looking at kind of the normal stages of development, like when the ears unfold, when the vaginal opening happens, when their eyes open, things like that. We also tested some reflexes to see kind of how that early development works in these animals to see if it was kind of on time. So this is an example of one of these tests. This is called the surface riding test, and it's basically a reflex. And for anybody who has a cat, you're going to completely understand this test. So essentially, we take these little baby pups and we pin them on their back, and we allow them, we track the amount of time it takes for them to want to roll over and flip onto their front. So that's a natural reflex that animals that walk on all fours don't want to be on their back. They'd rather be on the front. And so we can measure that amount of time. And importantly, this is a reflex that's tested before their eyes open, so they're not having a lot of fear associated with light and things like that when we're doing the test. So it kind of takes fear out of the equation. But it involves musculature, right? So it involves like trunk strength, coordinated movement, and it also involves vestibular system as far as the inner ear for orienting what's up and down. And so it takes kind of a number of different components into account. So in this case, we actually see not that the females are more affected in these early tests, but actually the males. So the males here take a significantly longer amount of time to roll over to the front from back, and which suggests to us, we only took one time point, but it suggests to us that they could either be delayed or that there's something going on with one of those systems that's involved in the writing. We also, in kind of conjunction with normal development, we also just took normal milestone measurements. So every time we did behavioral testing, we weighed the animals. At the end, before we sacrificed them, we took their body length and weighed their organs. So when we kind of look back at some of these data after the fact, we noticed that not only could we tell by eye or predict by eye probably who was a heterozygote because they were slightly smaller than their wild type litter mates, but when we started looking at the data, we found that that was true. So the body length measurements we took at the end, when we sacrificed them, we found that the heterozygotes in both sexes were shorter than the wild type litter mates. And we also found that the weights were lower as well. And that was across the entire lifespan that we measured. The males were more severe than the females in this case, but both were statistically significant. And interestingly, we went back into the second study where we took early animals, so around this P17 day, right here, where we do see there's a significant difference in body weight between the males. It is lower in females, but not individually significant at that time point. And we weighed organs just to kind of see early on if there was a difference in organ weight. And interestingly, what we found is that some of the organs were indeed, especially in the males, where there was a significant difference in body weight, the organs were also lower weight. So suggesting that it really kind of was a systemic issue. However, what was kind of interesting is the brains weren't a different weight at that age, which we would have expected. And in fact, when we normalized these weights to the body weight, the brain appears actually to be heavier than you'd expect for the weight of the animal. So this is kind of interesting, and we're trying to think about whether this might be or we could call it a relative macrocephaly phenotype, and whether there might be something that we can dig into a little bit more on this side of the house. So we decided to follow up with this body size thing a little bit more. And this was led kind of by a graduate student that I'll show you here in a minute, thinking about whether skeletal muscle might be involved in this whole equation. And that's for several reasons. And the first was really that there was a small literature a few years before we started our study that suggested that KMT5B might be involved in skeletal muscle development. And so this study was looking specifically at a type of muscular dystrophy called FSHD. And basically what they did in their model was they made a double knockout of KMT5B and KMT5C. So that's the one that does the trimethylation mark, specifically in skeletal muscle. And what they saw is that they knocked down the expression, they knocked down the trimethylation mark. So and that then was associated with increased necrotic skeletal muscle fibers. And so they were suggesting in this study in particular that could be representative of muscular dystrophy-like phenotype. There was another study a couple years after that that actually went a little bit further into this and further postulated or hypothesized and characterized this idea that KMT5B might actually be involved in muscle stem cell populations and maintaining their quiescence. So when your muscle gets damaged, your stem cell populations within the muscle get reactivated and basically build that muscle back up again, which is essentially the same thing that happens when you do weightlifting and build muscle mass, as well as those skeletal muscle stem cell populations are getting activated and you're increasing the size of your muscle fibers. And so they suggested that KMT5B might be involved in that switch process. So that was interesting, and it made us think a lot more about skeletal muscle. We also know that skeletal muscle contributes really significantly to total body weight. That's why they measure muscle mass and use that to calculate your total body fat. And so that made us think, well, is this basically this low weight that we see in our animals? Is it because KMT5B is somehow misregulating the development of skeletal muscle? And that also kind of leads into this idea of seeing some of those movement disorder, gene expression data sets come up in our gene expression data. So this little study was led by a graduate student in my lab, Jason Hewlin. He graduated last year and is now working at AbbVie in industry. It also contributed to this project. So Dorothy Kenny, who was an M1 summer research student, she's now in residency. She contributed to this project, and also Rebecca Black, who was an undergrad in the lab, really great, who's now in vet school at University of Minnesota. So for this study, we decided to look more specifically at skeletal muscle and whether muscle mass was changing or anything about the skeletal muscle physiology was changing. So we do see, based on some added behavioral tests that we did. So this one is a grip strength test. So we borrowed this grip strength meter from some folks in exercise science. Essentially, there's a little pull bar. This works really similarly to a luggage scale, essentially. You put the little mouse on, you pull them back by their tail, and when they release, it measures the maximal force that they could generate before they let go of the bar. And so that can essentially be used to measure force strength. So we do see that in adults, so these would be young adult animals, equivalent to young adult, that there is decreased force output from those animals in this test. It is very subtle, but it is statistically significant, but is not specifically driven by either sex. We see it instead as a kind of combined genotype relationship. So when we also, after that test, take the muscles out of these animals, we also wanted to weigh them, right? Because we had already looked at liver and brain and some of the other tissues to see if there's kind of this underweight, basically, phenotype in the heterozygous. So this is taking soleus and EDL muscles. Importantly, these are two muscles of the hind limb, lower limb, and they sit just adjacent to each other in the leg. The nice thing about them is they actually represent two different types of muscles. So soleus is a slow twitch muscle, EDL is a fast twitch muscle. So kind of in exercise physiology, these muscles are often used because they represent two populations of cell type that kind of sit right next to each other in the leg. And we do see overall that the weight of those muscles is significantly lower in both males and females in heterozygous animals. So again, suggesting there's something going on with size and acquisition of size of that tissue. So we wanted to look a bit about also kind of into the function of the muscle. So we looked at, just kind of the broad kind of characteristics, but wanted to see if there was anything going on at a kind of a more functional level. So one thing we looked at was innervation at the neuromuscular junction. So that's the junction where the motor neurons actually meet the skeletal muscle, where they can send signal for the muscle to have activity, right? So we can do this using some really nice and well-established immunofluorescence protocols. So in these protocols, we can stain the nerve with the green color here, and we can use basically another stain that's red to stain the muscle side of the neuromuscular junction. We can then look for overlap, so making sure that they actually overlap with each other, signifying a true and healthy interaction. And we can also look at the shape of them. So sometimes people call them pretzel structure, but essentially what we can do is use an ImageJ plugin to deconvolute this into a linear structure and track the linearity of this to kind of measure the health and the structure shape of the neuromuscular junction. So we call this continuity. So what we find is that there's increased discontinuity in our heterozygous muscles in males and females compared to the wild type, which suggests that there's perhaps not, there might be some structure function relationship that's causing some issues in the heterozygous state. The other thing we looked at was actually the muscle fibers themselves. So these muscle fibers are made of many individual myofibers, and the composition of the myofibers is how you can tell the difference between muscle types. So a slow twitch versus a fast twitch. So again, this was a protocol we did with immunofluorescence. We were able to do this all markers at once. So that we could actually quantify this for individual strands of muscle fiber. And so interestingly, what we found in this case was that there was no difference really in the distribution. So the numbers of myofibers in the right distribution were present in the muscles of the heterozygous animals. But the issue that we found is that in heterozygous animals, those myofibers were not as big as they should be. So to us, our final conclusion was that there was a hypertrophy defect. So everything's present in there, established, but it's not growing the way that it should. And we call this process hypertrophy. So that got us thinking about what might be causing this hypertrophy defect. And interestingly, through the literature, what we found is that factors called insulin growth factors, which are hormones, growth hormones, are actually regulate this process of muscular hypertrophy. So that's really interesting, right? So IGFs actually can bind to, there's IGF receptors and there's also insulin receptors. These can also bind the insulin receptors as well. And they signal downstream through IRS to two different pathways. So PI3 kinase, which is a growth pathway. So again, that makes sense why you see increased growth when you add IGFs into the system. And also the RAS-MAPK pathway. So when we read a little bit more about IGF, I got interested in it even more because there's really been some strong implications with autism. So we know, for example, that IGFs play a really actually very important role in the brain as well. So they're involved in neurostem cell division, neurogenesis, and synaptogenesis. So they're actually made by structures that sit in the ventricle, and they're used as a growth factor that basically bathes those neurostem cell populations that sit adjacent to the ventricle. So IGFs are really important there. And this is a family that includes two factors, IGF-1 and IGF-2. We also know that there are changes in circulating IGFs that have been identified among autism patients. So that's really interesting. We also know that IGFs can be administered systemically. They have been shown to improve cognition, learning, and memory. So that's sometimes in dementia models, but also in mouse models of autism. They also can improve social phenotypes in certain mouse models of autism. So that's exciting because it suggests that if it's administered systemically, but can have an effect on brain function, that it can cross the blood-brain barrier, which is really nice and often an issue when we talk about trying to find novel treatments for autism. And finally, IGF-1 actually is currently in a clinical trial for syndromic autism. And I've been in really active conversations with the clinical director of that clinical trial. So he works on Phelan-McDermott syndrome, which is a syndromic form of autism. And so we've been talking a lot about this and its potential and whether, we're talking about the same pathway, we're feeding in from a different side. We've been talking about this a fair bit. So that leads to kind of what our latest hypothesis is and kind of where my lab is going right now, which is this idea that KMT5B loss might result actually in a neuroendocrine disease that's characterized by these changes in IGF expression. And some of the kind of cool things about IGFs, or things that are cool and complicated, I guess, at the same time, is that IGFs are actually regulated by two different mechanisms. So there are endocrine IGFs that are produced. So these are the ones that you find in circulation. So they are driven primarily by growth hormone stimulation from the pituitary that signals to the liver to make IGF-1 and put it into circulation. So that is the bulk of what we would measure if we measure that in people and in mice as well, is the circulating or the endocrine form. But we also know from some evidence that isn't as well established that many tissues actually make their own paracrine or local expression of IGF as well. So for example, the mechanism I talked about in the brain, where we're stimulating those neural stem cells, that's mostly a paracrine mechanism. We also know that skeletal muscle makes its own IGF in a paracrine mechanism as well, and some of that can be put into circulation as well. So this is like a really complicated kind of crosstalk system that we are trying to understand about in our model. And I, leave it as a teaser, but, we are seeing that IGF is significantly different in our mouse model when we take samples over time. And we're also seeing that growth hormone, which is the hormone that's upstream of this, also might have some issues going on as well. So we do currently think that our hypothesis is holding water as far as that something's going on with the IGF signaling pathway.
And what do you think the source of the difference is?
Yeah, so that's what we're trying to track down right now. So we just ordered 2 new conditional models where we can knock out KMT5B in the brain and we can knock it out in the liver as well. So we're trying to test the endocrine mechanism by knocking it out in the liver and thinking more about the paracrine from the brain side of the house. But yeah, we don't know, and that's the kind of the fun chase. So that kind of leads us to, yeah, where we're at is that, how, where, and when does this regulate this process?
I mean, I wonder if you look like at the messenger RNA level, from brain and from liver, did that show anything?
We have those data, and it totally correlates with the so specifically the liver, the down regulation that we see circulating in our het animals. We see that similar level of down regulation in the mRNA in the liver as well. But that's a constitutive knockout. So we don't really know, if that's dependent on growth hormone stimulation. So there's also another intermediate called STAT5. That's a transcription factor required to drive the expression after growth hormone receptor activation. We have not looked at that. So that's one of the things we're thinking about too. So yeah, we're trying to understand because it's a constituent, right? KMT5B is down everywhere in our model right now until we have our specific conditional animals. So thinking about whether KMT5B is actually regulating the promoter of IGF in the liver, or if it's something upstream that has to do with the growth hormone expression in the brain, and whether that's coming from the brain and the neuroendocrine system, and kind of that feedback mechanism. Yeah.
A problem.
Yeah. It's a problem, but a good problem. Lots of questions. So that's it. I have to thank my lab, the people who did the work. Jody's an amazing mouse tech, and all the people who are in my lab right now, and then many who have left who worked on these projects. We get funding from many sources. So we've had some COBRI funding. We've been lucky, some internal funding, and we had a nice startup from Safari. And then our collaborators at Creighton, so the ones that were involved in this project specifically, Dr. Abel, and then 3 folks in exercise science who have been really great. So Kelly, Eric, and Jake have all been fantastic. And then my collaborators at CHOP, some of them have moved to NIH now. And then I always have to put out the plug since I also manage the biorepository here. So those folks didn't work on this project specifically, but are also another team that I manage that I didn't talk about all our cancer work and stuff today, but I'm always happy to if people want to do that offline. Thanks. That's it.
Awesome. Really interesting. I've seen your story at different points, and it's really nice to see how it's evolving. But I'll be quiet and let our audience unmute themselves and go ahead and jump in with questions.
I know we're a small audience, small but mighty.
Yeah. You get a huge compliment in the chat. Great presentation,
Thanks,
Dr. Stessman.
I know, I feel like this is the story I don't normally get to talk about.
Yeah.
Hey, Holly, this is Maureen Tierney. That was really fantastic. And as you know, both as a medical professional and as a mom, I'm particularly interested in your findings. To be honest, I'm not smart enough on the science part of this to actually ask a question. I just wanted to make a comment that I just find this fascinating, and I really want to watch along as with your work as you continue to sort of uncover some of this mystery.
Well,
Thank you for what you've done.
I really appreciate that. Yeah. And I know one of the things that so right now, our CHOP collaborators have decided that they're no longer, they actually were just kind of working on KMT5B as a side project, whereas it's my primary interest. So I'm actually moving the registry over here of these patients and trying to recruit more. So I was lucky enough that the private Facebook group that these patients are part of allowed me to be in their group. And many of them have approached me. So we are actively starting a KMT5B registry here. So that's been really great. We've had a lot of interest from the families, people reaching out. saying like, oh, I know I'm patient, whatever, and I want to talk about, so that's been really fun. And I think it's, one of the challenges for our field moving forward is to try to do a better job of, not treating patients as, a group of mice, right? And thinking about, do they want to participate in this research? Are they interested in it? What are the things that they want to know? So there's a big movement in our field to really integrate families into all of this and get the buy-in, and I think that will improve our science.
I want one relevant question for those families, and then there's a question in the chat too, but what's the frequency of this KMT5B